the inorganic, the organic and then the inorganic component as well. Then we'll look at the transport mechanisms in cells, where we'll look at both diffusion, we'll look at active transport, we'll look at passive transport. So under passive transport, we'll look at diffusion, osmosis, we'll look at active transport, we'll look at vesicle mediated transport as well. So this is what we'll be looking at in the next 2 hours. So I believe that we'll be able to pay rapt attention and follow as we go. So at the end of the presentation today, I expect you to be able to state one role performed by each of the substances that constitute cell inclusions. So we'll outline some component or some agastic substances. Then you should be able to look at some of the, So we should be able to look at the role performed by each of the substances. They will list examples of substances that form cell inclusions. Then we'll delve and look at the organic substances that are found in cell. We'll explain why the cell membrane is partially permeable. Then we'll look at diffusion, osmosis, active transport. Then we'll describe tonicity. And then we'll end up by looking at the channel and carrier proteins as well. So this is basically what we are doing. When there is time, we'll look at lactose intolerance, and then I have a table where I've summarized all the various movements on that particular table. So you'll be able to look at that table, and then we'll be able to appreciate where we see whether the substance is moving against a concentration gradient or moving along a concentration gradient, whether that particular mechanism involves energy or not, and whether that particular mechanism occurs through the plasma or through the phospholipid bilayer or through a gated protein channels. So we'll be looking at that today. Now, for the cell that we are looking, there are both living and non-living components of the cell. So there are living and non-living components of the cell. For the non-living component of the cell, so once you see the cell, something should tell you that no, though the cell is living, but the cell started from somewhere before it got to the living part that we see and appreciate as cell. So first of all, it starts from an atom. And who can help us define what an atom is from our chemistry class? Yes, one person to define what an atom is for us. Yes, anybody, just raise up your hand and define what an atom is. Okay, let's hear from Philemon.
An atom is the basic unit of an element that can take part in a chemical reaction.
Okay, thank you very much. So an atom is basically or is simply the basic unit of an element that can take part in what? In chemical reaction. So it means that when you see the cell has some component or it starts from an atom. Then we come to molecule. So what's a molecule? Yes, today we'll do a lot of talking. What's a molecule? So that it help you to revise your chemistry as well. What's a molecule? When we say a molecule, what comes to your mind? A molecule. When you talk of molecule, what comes to our mind? Yes, anybody to help us? Should I mention names? Anybody to help us? What's a molecule? A molecule. Anybody? Oh, nobody to help us with definition of a molecule. Okay. Okay, so let's hear. Let's hear from Abina. So Abina, what's a molecule?
A molecule is one or more atom. It's made up of one or more atoms.
Okay, so when different atoms come together, they form what you call what? A molecule. And from molecule, then we get to the organelles. So you could see how the cells or how the cell is arranged. So from atom, which is the basic unit of an element, then we come to molecule, which is made up of two or more elements or atoms. Then examples of the molecules are, we have carbohydrate and we have protein. So they are examples of molecules. Then from molecules, the molecules assemble or come together to form what we call the organelle. And you could agree with me that when we were looking at the organelles last week, we made mention of the component of each of the organelles. When you pick the example, you pick the cell membrane, you are able to detect that, okay, the cell membrane is made up of a phospholipid bilayer, where we have lipid in there, we have proteins in there, we have carbohydrates in there. So we are able to actually mention the various components of each of the various organelles that we studied last time. And these organelles are derived from molecules. So they are either carbohydrates, they are either proteins, some are lipid, or some are even glycoproteins. Are we getting it? So that is the non-living levels. Then from there, we move to the living level. So this is the living level of the organism. So when the organelles together come together, then they form what you call what? The cell. So we realize that the cell is simply what? The basic structural and functional unit of what? Life. Then from that level, we come to the tissue, which is a group of similar cells that are working together to perform the same function. Then we come to the organ, which is a group of tissues working together to perform a specific function. We come to organ system, which is what? A group of organs working together to form the same, performing the same function. Then we come to the organism, which is made up of what? The organ systems that are what? Comes all together. So this is the level of organization in multicellular organism. So we have the non-living level, then we come to the living what? Level. Please, any question on that? Any question on that? Are we okay? If you,
Yes.
if you are okay, just give me a thumb up, something, so that I know you are following. Are we okay? Can we continue? Okay, that's fine. So we should know that it starts from the non-living level, where you have the atom, we have the molecule, and then you come to the organelle. Then the organelles then transition into the living level, where we have the cell. You know, a cell is basically made up of organelles. Then from cell, we come to the tissue, come to the organ, we come to the organ system, then finally to what? The organism. So that is the composition of every multicellular organism. So you cannot say you are a multicellular organism and you don't have an element, an atom or an element in your system, or there are no molecules in your system, or there are no organelles. It doesn't work that way. So they all come together. Then we have the whole organism that we see. So you could see that it's quite a journey from the non-living transitioning to the what? Living level. So let's come to the cell inclusions. So they are non-living chemical component of the cell that constitutes the cell. So they are the non-living chemical component of the cell, and they are also known as the cell inclusions or the agastic substances. So you could really remember that we looked at the non-living component of the cell, where we made mention of some atom, some element, and then some molecules. So they are what is termed as what? The inclusion elements. So some refer to them as the agastic substances, others cell inclusions. They are simply these chemical components of the cell. So the major elements present are carbon, hydrogen, and oxygen. So as for carbon, hydrogen, and oxygen, mainly they are the major component of elements that are found in every cell or the organism. So so if you look at the protein, if you look at the structure, you are doing chemistry. So if you look at the structure of most of this protein or the carbohydrate, you will see that you will find carbon. For carbon, hydrogen, and oxygen, you are most likely to find them there. Before, if you are seeing some nitrogen imide group somewhere, you'll be able to say this is a protein or other molecules that are present, but major or majority of the elements that are present are carbon, hydrogen, and oxygen. And cell inclusions may be broadly divided into both organic and inorganic components. So the inclusion substances, we have both the organic and then the inorganic substances. So there are some substances that are organic and there are some substances that are inorganic. And all this forms part of the cell, or all these are part of the cell. And then we'll look at them one by one. And what I want you to understand is that each of them have a specific role they are playing. So we'll get to see some deficiencies, and you appreciate so much when we get to look at their deficiencies or when they are absent in the cell, then you get to understand the role they tend to play when they are absent. I believe you've done this over and over again, so to make the discussion very easier. So they exist in the soluble form in vacuoles and insoluble form in the cytoplasm. So when you see these cell inclusions, when they are present in the vacuole, they are soluble, because the vacuole, we have something we call the sap in the vacuole, and contains of soluble substances and water. Then in the cytoplasm, they are insoluble. So you see them both in the vacuole and the cytoplasm. Of course, they are all parts of the cell, but when they exist in the vacuole, they are soluble. When they exist in the cytoplasm, they are insoluble. Soluble simply means it dissolves in water or it dissolves in liquid. Insoluble means they do not dissolve in water. So examples are carbohydrate, you can mention them, glucose, lipids, protein, vitamins, nucleic acids, gums, resins, alkalis. All these are examples of inclusion bodies or gastric substances that are found in the cell. So we'll look at them. We'll not be able to treat all of them, but we'll look at the major ones that exist, both the organic and the inorganic ones. We'll look at them into details, and then we'll look at primarily the function that they play in the cell. So the first one we'll look at is we want to start with the organic compound. So let me ask this question. When we say an organic compound, what comes to mind? If we say organic compound from our chemistry class, what comes to mind? Organic compound. Organic compound. Okay, let's hear from Mafu. Okay, Mafu.
Okay, sir. They are compounds obtained from natural source, example from plants or animals.
Oh, okay, so they are obtained from natural source.
Sources, yes, sir.
Okay, that's. Can we get another view? Okay, Richmond, please.
Yes, sir. When we talk about organic compound, we are referring to carbon compounds.
Okay.
In the chemistry term, we refer to the carbon compounds. Yeah.
Oh, okay. So basically, when we talk of organic compound, as he rightly said, and as Mafu also rightly said, in chemistry, we want to look at compounds that have carbon, and these carbon are bonded to either hydrogen or other elements. So you can have bonded to hydrogen, you can have bonding to what? Either oxygen, nitrogen, sulfur, or some halogen. So that is basically what organic compounds are. So you have the carbon is there, and then the carbon is bonded to other elements. And these other elements, usually you find hydrogen, you find oxygen, then you find, in some instances, you find nitrogen, you find sulfur, and then you find other halogens as well. So that is basically what organic compounds are. So the first one we want to look at is carbohydrates. Carbohydrates. So basically, we all know carbohydrate. Carbohydrate. We've been discussing carbohydrate. Carbohydrate is an organic compound, and they are mainly composed of carbon, hydrogen, and oxygen. So chemically, I should have brought some chemical structure of a carbohydrate here. It's escaping, but I'll make sure I'll add it before I send the slides to you. So you could actually appreciate the structure of a carbohydrate. You see that basically carbohydrate, you find carbon in there, you find hydrogen there, and you find oxygen there. And carbohydrates is mainly divided into, or we have groups. So we have the monosaccharides, we have the disaccharides, and then we have the polysaccharides. When we talk of monosaccharides, it means that they are composed of what? A single unit. Mono means what? One. Di means two. Poly means what? More. Are we getting it? So when we talk of monosaccharide, examples are, we have the glucose, we have the fructose, and then we have the what? The galactose. I didn't bring galactose, but galactose is also a monosaccharide. So they are the simplest form of the sugars. Are we getting it? So the simple form of the carbohydrate, or the basic. So you say they are the basic, the simplest form of the carbohydrate is the monosaccharide, because they are made up of one sugar unit, single sugar unit. I hope we are getting it. So this is not something we should do chew and pour. But when you see mono should give you an idea that it's one. Di is what? Two, and then poly is what? More or many. So monosaccharide, and examples are the glucose, the fructose, and the galactose. Then we have the disaccharides, where we have the sucrose, then we have the maltose, then we have the polysaccharide. We are much more interested in the polysaccharide because that's mainly concept. So you can see your cellulose here. When you hear cellulose, what comes to our mind? Can be composition of the plant cell wall. Then we have starch. Starch is the plant stores their excess glucose in a form of starch. Plant stores their excess glucose in the form of starch. The animal stores their excess in the form of what? Glycogen. Then we come to lignin. I believe we looked at lignin last week. So we should remember lignin, and then how they confer structural integrity to the cell as well. Then we come to chitin, which is the structural component of the cell wall of the fungi. So this is basically for the carbohydrate. So starch in plants and glycogen in animals. So you could see that you will not hear any scientist telling you that there is starch in an animal. No, because animals do not store their carbohydrates in the form of starch. They store it in the form of glycogen. It's plants that stores in the form of what? Starch. Then mainly the function of carbohydrate mainly is energy supply, and that is glucose. So you could look at how we get our energy sources, while the carbohydrate we take in is broken down to glucose. Glycolysis takes place, Krebs cycle, and then the electron transport chain, where at the end we get ATP, which is the principal energy molecule. So that is energy supply, glucose. Then the structural component in plant and animal tissue. So we'll look at cellulose in plant, chitin in what? In fungi. Then we can look at the which other one? The peptidoglycan in the bacteria. So that is basically well. So let's take notice of the good. Any question on carbohydrate? Can we proceed? Okay. Then the next one we want to look at is the protein. The protein, yes, is an organic compound, and they are made up of. So look at the composition of protein. When we're looking at carbohydrates, we mentioned of carbon, hydrogen, and oxygen. But with protein, they are made up of carbon, hydrogen, oxygen, and nitrogen. So carbon, hydrogen, oxygen, and nitrogen. Some contain sulfur and phosphorus as well. So some may contain sulfur and phosphorus in some instances. So they are formed from smaller units called the amino acids. So just like the glucose is the basic, or the monosaccharides are the basic, the smaller units are known as amino acids. So the amino acids come together with peptide bonds, and then they form what you call the polypeptide, which is the protein. Proteins are important structural component of the protoplasm. So we cannot do away with proteins. Enzymes which catalyze most metabolic activities are made up of proteins. So enzymes are protein in nature. That is why some instances when you apply some level of heat or small, you tend to denature the protein, or you tend to denature the enzyme. So enzymes, or like other proteins, are able to work very well at the optimal temperatures. When you tend to hot or when you tend to change this temperature, then you tend to denature these proteins or the enzymes. So they are important for growth and repair of body tissues. We all know the functions of protein. And so let's do a bit of nutrition. What do you think will happen to somebody who doesn't take protein in their diets? Yes, anybody. What do you think will happen to somebody who does not take protein in their diet? Yes, let's reach. Okay. I've not heard from
Yes,
Philemon today, so let me hear from Philemon Richmond.
okay.
Okay, Philemon, let's hear from you.
Okay, as someone who is protein deficient will suffer from kwashiorkor.
So a very clear and very common one we know is kwashiorkor. So that's the so protein plays an important role in the system of every organism. So they are important for growth and repair of body tissues. Definitely, there will be some of the cells, some of the tissues will be worn out. Definitely, there will be injuries. Definitely, there will be wear and tear. So how does the body tend to replace some of these things that happen? So it is as a result of this protein. So it's proteins play an important role. And proteins also have a skeletal or protective function. For example, keratin, collagen, myosin, and actin. I'm sure we'll look at this in detail somewhere next week or in our lecture for where we'll tend to look at the muscle tissue. And then we'll look at the skeletal tissues as well. Then we'll look at some of these protein compounds and the role that they tend to play in the skeletal system as well. So protein is very, very important. So those of us who tend to eat diet without protein, we should know that protein have a lot of functions they play. So we should normalize adding proteins to our diet. So that one we'll look at is lipids. So when we talk of lipids, we are simply looking at fat and oil. What is the difference between fat and oil? Yes, anybody? What's the difference between fat and oil? Yeah. Hello, Richmond.
Yes, sir. The difference between fat and oil is the fats are obtained from plant, and as well, oil is obtained from plants. And another one that we can use to differentiate is fat is
No, please come
solid at
again.
room
Come
temperature.
again. Your first difference. Come again.
The fat is obtained from animal, and the
Okay.
oil is obtained from plants. And
Okay.
another difference is the fat is solid at room temperature, and oil is liquid at room temperature. So that is what
Oh,
I can say.
okay. Okay. Emmanuel, do you want to add something? Emmanuel, okay. Emmanuel, can we hear from you? Okay, it seems Emmanuel has network challenge. So let's hear from Eric.
Surface fat has more saturated in them, whilst oil have unsaturated in them.
Oh, okay. Okay, so that's fine. So at room temperature, fat is solid, and at room temperature, oil is what? It's liquid. So that is basically. So these contain carbon, hydrogen, and oxygen, but with less proportion of what? Oxygen. So you can see that carbohydrates also have carbon, hydrogen, and oxygen. But with fat, there is what? Less proportion of what? Of the oxygen. It means that the quantum of oxygen molecules that you found in carbohydrate will be more than those that you find in lipids. I believe that is clear. When oxidized, the molecule of fat generates twice as much energy as an equal amount of carbohydrate. So this is a serious statement. When oxidized, the molecule of fat generates twice as much energy as that equal to carbohydrate. So what it means is that there are some instances where you don't have or you've not taken in carbohydrate molecule. But in the absence of carbohydrate, fat plays the role of energy production. And when we look at glycolysis and Krebs cycle, we'll look at some elements that fits into these various cycles. And there are instances where even protein serves as a substrate for glycolysis or for energy production. So we'll look at that into details when we get there. But what I want you to know is that it's not only carbohydrate that produces energy. Fat produces a lot of energy. And there are instances where when carbohydrate is absent, fat is absent. Even protein can enter into the glycolysis and then produce some level of ATP. So that is basically about that. Fat is chemically inert and so can be stored under the skin and around the viscera. So you see that under the skin there's fat, there's adipose tissue. So we'll look at adipose tissue somewhere next week when we are looking at the connective tissue, where that is deposit of fat. Then we'll look at the viscera. So most of these organs are surrounded by fat because fat plays a critical role. It protects the organs, it insulates them as well. You know how temperature varies. So it is this fat that insulates the body against either cold or heat loss. So those of us who undergo a lot of butcher works or we tend to kill animals, you realize that when you slaughter your goat in your house, you realize that you could see that most of the delicate organs, you see some of these fats that are around them, and they have an important role. Even below the skin, they have an importance. There are some animals that their fats are highly saturated, and then there are some animals that their fat are not so saturated. I believe you can read further on that to know some animals have fats that are not saturated. Others, their fats are highly, highly saturated. And you can read on that. So they insulate the body against cold or heat, as I rightly said. Plants store fat as oil in their seeds. This oil may be oxidized to provide water for germination or extracted for others. So as we rightly said, there are some, so you could ask yourself, why is it that there are some instances where there is no water that is available, but the seeds germinate? So it's as a result of oxidation of fat. So far as there is hydrogen, there is oxygen inside, it means that those two elements can be oxidized and the water can be produced. I believe you are getting it. There's hydrogen and there's oxygen, and you know water is H2O. It means that they can be oxidized to produce water for germination or extracted for other purposes. So let's have that in mind, that when you see seed germinating and there is no water, it doesn't necessarily mean that water is an important component needed for germination. But the fat oil can also be oxidized, and once water is produced and the seed is viable and the soil is okay, germination will take place, even without you necessarily pouring water on it. Or then we look at vitamins. So there are a lot of vitamins. We can speak about vitamins. We have the fat-soluble vitamins, and then we have the water-soluble vitamins. Can we mention the fat-soluble vitamins? The fat-soluble vitamins. Can we mention them? The fat-soluble ones. Yes. There are 4 of them, the fat-soluble vitamins and then the water-soluble vitamins. Do you remember them? From, okay, Michael.
Hello, sir. So the fat-soluble vitamin, I know of vitamin A, retinol, vitamin D, calciferol, vitamin E, and vitamin K.
Okay, so that's fine. So vitamin A, vitamin D, vitamin E, and vitamin K are the fat-soluble vitamins, and vitamin B and C are the water-soluble vitamins. So you could see that I just summarized everything simple here. But you realize that when you take vitamin A, there are functions of vitamin A, there are roles vitamin A play, and then deficiency in vitamin A. So when you take vitamin A, you should be able to tell me the food sources. What foods can we get vitamin A from? So let's let's look at vitamin A. What are some of the food sources that we can get vitamin A from? Yes. Okay, Philemon.
Vitamin A can be gotten from oil and then bread as well.
Oil and bread, vitamin A. So what is in the bread that is giving the vitamin? Okay, yes, Emmanuel.
Can get some from mango.
Mango.
Yeah, mango, carrots, spinach. Those kind of fruits, we can get
So
vitamin A from there.
Mainly we get vitamin A from those green leafy vegetables.
And
See your
liver.
green,
Yes, sir.
yes, your green leafy vegetables. You mostly get vitamin A from them. Yes, Eric.
And simply, can we get egg? I think egg too is part.
Egg. You get vitamin A from egg.
No.
Yes, please.
But I
Hello?
said egg, I think it's protein.
Yes.
I know it's protein, but well,
Okay, the Abner sound is up.
From palm oil. We can get vitamin A.
You said consume.
And then
Yes,
from
yes.
palm oil.
So that is the so those green leafy vegetables, yes, you can get some proportion from egg as well. So that is the vitamin A. And what do you think are the one of the key importance of vitamin A? Mercy, please mute your microphone. Mercy, kindly mute your microphone. Okay, what's one key importance of vitamin A? Okay, let me hear from let me hear from Michael.
Okay, sir. So because of the presence of the green leafy vegetables, it provides iron.
Eric, I'm coming. Eric, please mute your microphone. Eric and Richmond. Okay, please go on, Michael.
Yeah, please. I said because of the presence of the green leafy vegetables that we take, we get iron from it, which is very important for the body growth.
Okay. Let's hear from Abina.
It also helps for clear vision with the eyes.
Yeah, so vision. Okay, Richmond.
Oh, sir. It also helps to boost the immune system.
To boost the immune system.
It helps the white blood cell, or it support the white blood cell to fight against diseases.
So that's it helps in immune response. Okay, so that is, and then so from what you could say, so it means that when you are deficient in vitamin A, you are most likely to have problems with your vision or your sight. Are we getting it? Yeah, so you could see that vitamins, though they are taken in very small quantities, they have huge role they play in the body. And if you mess up with them and you are deficient in any of them, you could see that it has a lot of serious consequences. So we can do that for vitamin A, vitamin B, vitamin C, D, E, and what? And K. So they are responsible for healthy growth of the body, as we rightly said. Though they are required in small amounts, deficiency in any of them has a profound effect on the normal function of the body. So you see that when you are deficient in any of these vitamins, you could see that it has a lot of effect on the body. So for example, scurvy is associated with lack of vitamin C, and beriberi is caused by lack of vitamin B. So beriberi is what you call pocket lawyer in the male. So you see gentlemen dress very well and you see their hand in their pocket and they are scratching somewhere. So they lack. So it's sometimes as a result of lack of vitamin B, and when we are suffering from this beriberi, so we have to take our vitamins very. Though they are required in small amount, but if you lack any of them, it's difficult. Then we come to the nucleic acids. So for nucleic acids, we know that there are two types of nucleic acid has to do with the DNA and RNA. And I believe basic genetics and cytology, you've done a lot about the DNA and then the RNA. So I don't need to go so much deep into that. They consist of the unit called the nucleotides. So we know of the nucleotides, where the nucleotides consist of what? The nucleosides. So we have the nucleosides and then the phosphate group as well. So we have the nucleotide, that is the nitrogenous bases. Then you have your phosphate group. Then you have what? There are three. So I believe you remember. Then they consist of unit called nucleotide. The DNA is restricted to the nucleus. So as for the DNA, it's only found in the nucleus. That is in the eukaryotes. So the DNA is found strictly in the nucleus. But with the prokaryotes, they don't have nucleus. So their genetic material is just scattered in the cytoplasm. But the RNA is found in both the nucleus and the cytoplasm, but mostly in the cytoplasm. So the RNA, we have three types. We have the messenger RNA, we have the ribosomal RNA, and we have the transport RNA. Now, the messenger RNA, which is the mRNA, is found partly in the nucleus. So it goes into the nucleus, or it's in the nucleus, copies the genetic information, then immediately it moves through the nuclear pores into the cytoplasm, where it's being transcribed and translated into what? Into protein. That's why I say that the RNA is found in both the nucleus and the cytoplasm. So the messenger RNA is found in the nucleus, but once it copies the genetic information, once it is transcribed, it moves into the cytoplasm, where it undergoes the translation and then it is translated into a specific protein. So that's why we have it in both the nucleus. You read some books and they will tell you that nucleus is found, DNA is found in the nucleus, RNA is found in the cytoplasm. It is not always the case. There are some instances where the RNA can be found in the nucleus, and that only happens in the messenger RNA when it's copying the genetic information from the DNA for translation to take place. So let's take notice of that. Nucleic acids are associated with inheritance of characteristics and synthesis of protein. So inheritance, so the gene, what you pass on to your next generations, are all associated with your nucleic acid. So you could see that when there's any defect in any of the genes or any of these nucleic acid, it has serious consequences. I believe you looked at mutation, where you look at any alteration in just one mutation, 1 point mutation, then you could see the effect that it has on. You've seen sickle cell anemia, and you've seen just a defect in one of the gene point mutation, and you see a whole lot of situations. So that is basically about the nucleic acid. Any question on the organic compounds? Any question on the organic compound? Any question? Okay, so we can move on. Then we come to the inorganic component. So with the inorganic compound, the first one we'll look at is water. Water. So what is the chemical composition of water? Yes, anybody to help us? Chemical composition of water. Okay, Prince.
Hydrogen and oxygen.
So chemical composition is hydrogen and what? Oxygen. And you realize that in most organisms, most of their protoplasm is composed of water. So water constitutes majority of the weight of the organism. So it is the most abundant compound in the cell, constituting between 65 to 90 by weight of the protoplasm. So if you have the organism or the organ or the whole system, and you have about 65 to 90% composing of water, then you could see that then its majority of the weight of the protoplasm is coming from water. But even it's only 65, it's more than 50%. And some travels as much as 90%. So that should explain to you the importance of water. Water is a solvent in which other compounds of the protoplasm are dissolved. Remember, we said when we're looking at the cell inclusion, we said some are organic, some are inorganic, and there are some that are soluble in water and others that are not soluble in water. And we said those that are soluble in water are found in the vacuole, and those that are not soluble are found in the cytoplasm. So for most of these compounds, water is the solvent. So we'll look at solutions as we go on. It serves as the medium in which chemical reactions in the cell takes place. For example, respiration, photosynthesis. All these are important reactions or processes that takes place in the cell. And without respiration, I don't think we will be able to survive, because where will you get your energy from? Without photosynthesis, where would we get our carbon source from? So these are very important processes that require water. Then we look at gases. So we can also look at the importance of gases to every organism. So oxygen and carbon dioxide are the most common gases in the cell. So one is taken in, one is taken out. So oxygen is used in the process of respiration to release energy from food. So even with respiration, we have types. We'll look at that in detail, but we have aerobic and anaerobic respiration, where we have one that takes place in the presence of oxygen and another taking place in the absence of oxygen. But mostly or mainly, it takes place in the presence of, so majority of respiration takes place in the presence of oxygen, unless there are organisms that are strictly anaerobes, and they do everything in the absence of oxygen. Example has to do with the Clostridium tetani. That is the bacteria that causes tetanus. They are strictly anaerobe, and so they work or they operate well in areas where there is no oxygen. So that is some. And then we also have some organisms that are microaerophilic. They require just small amounts of oxygen to thrive. Then we have the aerobes, those that requires oxygen, anaerobes, those that do not require oxygen. All these are terminologies that comes with gases. So carbon dioxide is generated as a waste product after the process of respiration. So after respiration, you see carbon dioxide that is generated as waste. By this happens in plants, in animals, we bring out carbon dioxide, and then the plant takes up the carbon dioxide for photosynthesis. But this happens during the day. During the night, plants rather take in oxygen. Can somebody tell us why? During the day, plant takes in carbon dioxide and they bring out oxygen, but during the night, they take in oxygen. Can anybody explain to us why? Or I should give it to you as reading assignment. Are we there? I just want to be sure if we are not sleeping. Okay. Eric. Any contribution? Eric.
Sir, sir, I think it's due to photosynthesis.
Photosynthesis when?
During the day, the carbon dioxide is being used to manufacture the food by the plants.
Oh, okay.
And in the night,
During
the night, they make use of the food, so there is no need for them to be getting carbon dioxide during the night.
Okay, so it means that during the night there's no light. So it means that they will not be able to undergo photosynthesis. Yes. So then they have to make use of the food. They have to make use of the food that they've manufactured and break down that food through respiration, and that requires oxygen. So in the night they take in oxygen. So that's okay. So the different concentration of gases in the cell enable diffusion between the cell and its environment to take place. So we'll look at diffusion in details, where we'll look at differences in concentration gradients. We get to understand that molecules will only move when there are differences in concentration gradient. If there's no difference in concentration gradient, there's no way molecules are going to move. When one level is higher than the other, then you see molecules that tends to what? To move. There are some instances where molecules will tend to move against concentration gradient. I think that are something that we can look at. Then we come to the salt. So salt, we have sodium chloride, which is a soluble salt and which ionizes in water to form ions. Then the dissolved ions make the cytoplasm a good conductor of electricity. The current is generated as a result of ions moving in solution. This property is important in image such as nerve cells. So when we look at the nervous cell or the nervous tissue, you get to appreciate how these ions take place. And even when we look at co-transport, we'll look at co-transport and active transport in some few minutes. So you get to understand the role some of these ions play in facilitating movement, as well as in the nervous system. So nervous system has to do with the system that is responsible for transmitting impulses, transmitting signals and action. So the nerve cells mainly depend on this ion. So differences in ion concentration is what causes it. So I'm sure in our group presentation, one group will be presenting on some of these action potential and how the nerve carries these impulses. So when we get there, we'll look at that. So transport through cell membrane. Remember, we said that cell membrane is selectively permeable. The cell membrane allows some molecules to pass through and prevent other molecules to pass through. Now, the cell membrane has what we call the phospholipid bilayer, as well as it has some protein channel gates. So in the next hour, I want to look at the various ways by which molecules move across the plasma membrane. So the plasma membrane exhibits selective or partial permeability. It's the non-polar molecules, including hydrocarbons, carbon dioxide, and oxygen, are hydrophobic. When we talk of hydrophobic, it means that they hate water and easily dissolve in the lipid bilayer of the membrane and cross it easily. Ions and polar molecules move through protein channels such as channel proteins and carrier protein. So now, there are some molecules that easily pass through the phospholipid bilayer. They find themselves pass through and then they enter, depending on the molecular size of the molecule. And then there are some molecules that they do not pass through the phospholipid bilayer, but there are some protein channel gates. So there are some channels that those bigger molecules are able to pass through to the cell or exit the cell. Then there are some of them too that they need carrier protein. Some of them, based on their shape and based on their structure, they are unable to pass through either the phospholipid bilayer or the protein gate. So they need some carrier proteins to enable them to be able to pass through this. So we'll look at some of these molecules and then we'll look at how these processes takes place. So there are four basic mechanisms. We have diffusion and facilitated diffusion. We have osmosis, we have active transport, and then we have bulk transport. So diffusion, facilitated diffusion, osmosis, active transport, and then we'll look at bulk transport. So when we talk of diffusion, what is diffusion? When we talk of diffusion, what comes to our mind? Yes, anybody? When we talk of diffusion, what comes to your mind? I think this is a kind of revision. Diffusion. So let me mention names. Let me look through the class list. There are some people I've never heard their voice before. So Evelyn, what is diffusion? Evelyn, what is diffusion? Do you have Evelyn here? Evelyn, are you here? Okay, let's hear from, I think, Samuel. What is diffusion? Okay, so remember, as part of this program, your participation in class also carries marks. And so if you are online and I mention your name and you don't answer or you don't respond, what you are telling me is that you are not here. And that also has its own. Okay, let's hear from Marfu.
Okay, sir. Diffusion simply means the uniform distribution of molecules through fluids, either happening in liquid or in gas. That is what I think I can say. The evenly distribution of what? Molecules, either through liquids or gas. That's what I can say.
Oh, okay. Yes, let's hear from Enoch. Enoch.
Okay, sir, thank you. Is the movement of particles from the region of higher concentration to the region of lower concentration.
So is the movement of molecules from a region of what?
Higher concentration to the region of lower concentration.
Oh, okay. So diffusion is the movement of molecules from a region of higher concentration to a region of what? Lower concentration. And there's one thing we tend to forget. When the movement takes place, the movement always ends when there's what? Evenly distribution of the molecules. So until there's what? Even, or until the molecules are what? Evenly what? Distributed. So now let's look at two major things that comes into play when we are looking at movement. So we have solute. So the solute is the substance that is being dissolved. Then we have the solvent, that is the substance that is doing the dissolving, or the substance the solute is what is dissolving. So whenever you have a solution, you have both a solute and a solvent. You have a solution, you have a solute and a solvent. So the substance that is being dissolved is the solute, and then the solvent has to do with the substance that is being is doing what? The dissolution. So when you take, let's say, sugar solution, you realize that the solute is the sugar, then the solvent is what? Is the water. So that is basically about solute and solvent. So we'll look at passive. Remember, I was saying that there are two types of movement. Either it is passive or it is what? Active. Now, passive has to do with the movement of substances down a gradient across a membrane without energy. So any movement that has to do with the movement that does not require oxygen and it is down a concentration gradient is known as passive transport. So when the movement is taking place, it does not require energy and the movement is going down a concentration gradient. Enoch, your hand is up. Or it's a legacy hand.
Yes, sir. Legacy one, I will remove it.
Okay, okay. Thank you. It means that it is past. Yes.
Hello, sir. Yes, sir. I want to ask for the gradient, the meaning of it, as it is in the definition. I actually don't get it. So
you want to understand what gradient is. So gradient has to do with differences in concentration. Are you getting it? So if, for instance, you have 10 moles of a solution, one solution has a concentration of, let's say, 10 moles of a solute, another has 5 moles. Which one has a higher concentration? definitely the 10 moles, right? Yeah, so the gradient has to do with, you remember gradient. Gradient from mathematics means what? Slope. Are you getting it? So it has to do with differences in the concentration. Yeah, so gradient also means slope. And in slope, definitely one is higher than what? Than the other. Are we getting it? Yes. So the movement of substances down a gradient across a membrane without energy. All right. So examples of passive transport are diffusion, osmosis, and facilitated diffusion. Somebody will ask, what's the difference between diffusion and facilitated diffusion? So when you talk of diffusion, it means that the molecules is moving across the phospholipid bilayer without anything aiding it. Of the facilitated diffusion, the molecules are moving down a concentration gradient, all right, but their movement is being aided by either the gated channel or by the carrier protein. So though they are moving from a higher concentration to a lower concentration, their movement is being facilitated by either the gated channel or by a carrier protein. But with the diffusion, that is when the molecules freely move through the phospholipid bilayer from one end to the other. So that's it. And factors that determine passive transport include concentration gradient, temperature, molecular size, and medium or membrane permeability. So we'll look at that in the subsequent slide. So as we rightly said, diffusion has to do with the net movement of molecules from a region of high concentration to a region of lower concentration until a uniform mixture is attained. So the molecules move down a concentration gradient. So when you are moving down, it means that you are from the higher concentration to what? To a lower what? Concentration. The molecules have kinetic energy. When you talk of kinetic energy, it is the energy that causes molecules to move. So it is this kinetic energy that makes them to what? To move. As a result, diffusion molecules reach an equilibrium where they are evenly spread out. So the movement will be taking place. So the movement, so for instance, if you stand at one end of your room and you apply a perfume, you realize that at that particular point where you apply the perfume, the concentration is very what? Strong, or the concentration there is very high. But with time, you realize that the perfume will spread, will continue to spread across the whole room, and with time you will see that the whole room will have the same level of fragrance of the perfume that you spread out, and that is the movement. So until an equilibrium is when the molecules on A is equal to the molecules on what? On B. This is when there's no net movement of molecules from either side. So if you have two sides, and assuming the concentration here is 5 mole, the concentration here is 5 moles, the concentrations are the same, and so no, there will be no movement. But immediately one is higher than the other, then you see that there is some level of slope, and when there is slope, there will be movement. So you could bear with me that when our plumbers are doing plumbing work, always they leave some level of sloping, because when there is no slope, water is definitely not going to move, or the speed or the rate at which the water will move, unless you apply some level of pump or pressure. But so far as there is slope, you will see that the water will easily move even without pump. That is why your water tank on top of your house, you don't need a pump for the water to be distributed to the various rooms in the house, so far as it is at what? At the top, because at the top it has a huge difference in concentration between that and those that are there down, so it will easily flow to the bottom without any pump. But when you are moving the water from the ground to the tank at the top, that one you are moving it against concentration gradient, and you have to need energy, and that energy is what we term as what? Your pump in the case of the water systems. So let's look at this image. Can we see the image? What can we say about? Let's focus on the So the females, kindly help us. Is this red, or orange? Is it red or orange? Okay, Abina.
So it's red.
Okay, so the females have agreed that it is red. I know for the males we are color blind. We only know primary colors. So now we will see that this is inside the cell, and inside the cell we have. So let's focus on the red balls, the small red balls. So you realize that we are looking at the cell membrane. So this is the cell membrane. So the cell membrane is what is partitioning the inside of the cell from the outside of the cell. So you realize that the molecules are going to move. So let's count and see. So how many red balls do we have here inside the cell? Yes, can somebody count and give us a figure? Okay, Prince.
Sir, the 11.
They are 11. Okay, so the red balls are 11. Okay, so let's see. So can we see diffusion taking place? Are we following the image?
Yes,
Can we
sir.
see the diffusion taking place?
Yes, sir.
Yes. So you could see then. Can we see the final? So you could see that at the end, this has how many? 1, 2, 3, 4, 5, and this one has 1, 2, 3, 4, 5, 6. Of course, it is 11. So far as it is 11, definitely one there will be difference of one. So, but if it were to be 12, you could see that you have 6 here and 6 here. So at this point, we'll say that it is in what? Equilibrium. And at this point, there is no net movement of molecules because those that are here are equal or equivalent to those that are what? Here. Are we getting? Those that are inside the cell are equivalent to those that are outside the cell. So this is how diffusion takes place. So the movement will be taking place until it reaches an equilibrium or until the molecules are uniformly distributed. That is why you cannot define diffusion without bringing equilibrium or uniformity in distribution of the molecules. If I'm marking your paper and you just define movement of molecules from high concentration to low concentration and you leave it, I'll give you half. Because what you are telling me is that then they will just be moving, this one will be moving, this one will be moving. But no, when they reaches an equilibrium, which they will definitely get, when they get to an equilibrium, at that point, there's no net movement. So let's clearly take it. So let's look at some of the factors that affect the rate of diffusion. Remember, we said with passive movement or passive transport, there are definitely some factors that affect the movement of the molecules. So we'll look at them one after the other. So the first one that we'll look at is the steepness or the differences in concentration gradient. So you realize that the bigger the difference in concentration gradient, the quicker the rate of diffusion. So I always explain this point with the water system because that's something simple you can explain. So you realize that if you have a water hose that is on the same level, you realize that movement of water is a bit difficult. But when there's difference, when one point is higher than the other, you realize that the water easily flow without you even applying some level of force. And this difference in concentration gradient applies in most construction firms. So you realize that when they are even building roads or when they are constructing roads, they try to leave out these differences in concentration gradient, what we call the slope or the gradient, as well as even in constructing gutters, our plumbing or our drainage systems, all of them use this steepness because when one is steeper than the other, it means that definitely water is going to flow freely without any pressure or any force. So the bigger the difference between the two sides, the quicker the rate of diffusion. So when the concentrating gradients are very huge or very wide, it means that molecules are going to move very fast. But when the concentration is not so wide, it means that the movement is a bit going to, though the molecules are going to move, just that the pace will be. Then temperature. So you could bear with me that temperature increases the rate of diffusion. So you can just perform this experiment quickly in your homes. Try to have two solutions. Put let's say salt into a cold water and put salt into warm water. Which of them will dissolve faster? Yes. Okay, Michael.
Sir, please, the warm water.
The warm water. Enoch, can you tell us why the warm water will dissolve faster?
Because the temperature is very high. That will cause
Yes.
the molecule to move very fast or melt very fast.
It's as simple as that. Philemon, you have something to add?
Yes, please. The warm water, because the temperature is going to break the bond between the salt crystal, therefore increasing the movement of the molecules of all the salt, thereby fastering the dissolution of what the salt in the warm water compared to that of the cold water.
So it's as simple as that. Once you apply temperature, you increase kinetic energy. And once there's high kinetic energy, it means that molecules are going to move very fast. And once they are moving, they are going to dissolve very faster. So high temperature gives molecules or ions more kinetic energy. So molecules move around faster. So diffusion is very fast. So when you apply temperature, you realize that the rate at which molecules dissolves is very, very quick. So you can actually try, this is very practical. You can try it in your home this evening, and then you realize that just have a cold water, have a warm water, put water inside, just allow them the same quantity of salt, the same quantity of water. Then you realize that the warm water, the one that is warm, or hot, is going to dissolve faster than the one that's cold. It's just a result of kinetic energy movement of the molecules. Then the third one we'll look at is the surface area. So we'll look at the surface area. The greater the surface area, the faster the rate the diffusion can take place. Yes, anybody to explain that? Forget about my explanation that I gave. Use a layman definition to explain the greater the surface area, the faster the diffusion can take place. Yes, anybody, just a layman explanation to that. How will you explain that to your younger sister in the house? That the greater the surface area, the faster the diffusion can take place. Yes, anybody? Is Evelyn here? Okay, Enoch.
So thank you. So I think there will be more space for the free movement of the particles.
Exactly. So once there's more space, what will happen? If the surface area is wide, it means that there will be more space. So once there's more space, it means that kinetic energy, there will be more room for movement. And so the molecules are going to what? Dissolve very, very what? Or diffuse very fast. So you can liken these two. Do we remember the molecular structure of solid, liquids, and gas? Do we remember them? The molecular structure.
Yes, sir.
Yes.
Yes, you remember them, right? So you could see how molecules
Yes,
are
sir.
distributed in solids, how molecules are distributed in liquids, and how molecules are distributed in gases. You could see. So just take
Yes,
these
sir.
three. Let's use these three molecules as a case study: solid, liquid, and gases. Which of them do you think molecules will diffuse very fast in them, and why? Just raise up your hand and I'll call you. Use the molecular structure of the various solid, liquids, and gas, and let's see which of them you think molecules are going to diffuse very fast in them and why. Okay, let's hear from Michael.
Okay, so among the three states, the one that
So
diffusion.
so you speak on one. There are three hands up, so you speak on one. Yes.
I'll talk about the gas one because there are a lot of spaces within it. Hence, kinetic energy cause higher diffusion power as compared to the other two.
Okay. Okay. So that is fine. So Abina, which one do you want to speak on?
So please, I wanted to talk on the gas.
So Michael has already. So just you can do water, you can do solids. Any of them.
Okay. With the liquid, their particles are a bit freer than the solid, so it will also help with the diffusion.
Yes, but not as compared to gases where the molecules are
Yes.
widely distributed. Then you come to solid. You see, solid, the molecules are tight, very close, and so diffusion in solid is very. It's not impossible, but it's very, if you are in, it's very slower because of how the molecules are tightly packed in solid. So that has to do with the surface area as well. Now you look at the type of molecule that is diffusing. I get so the type of molecule that is. So you could bear with me that when the molecule is very large, definitely the molecule that is smaller will be able to diffuse very faster than a very larger molecule. I hope we will get that. Though there are some exceptions, but in normal real terms, the molecules that are smaller are going to diffuse faster than molecules that are very large. And also, we can look at the polarity of the molecules as well. So non-polar molecules diffuse more easily than polar molecules because they are soluble in the non-polar phospholipid tails. So you see how, do you remember how the structure is the phospholipid bilayer? So if the molecule is non-polar, it means that it's more likely to bind to the tails. And once it's able to go through the tails, it's able to either enter or exit using the phospholipid bilayer. Then another factor I want you to consider is stirring. Remember stirring. So you will see that stirring also increases the rate of diffusion. So you can actually practically do that when you have a solution, both water, let's say salt, and in one of them you add and you stir, in the other you add and you leave. You realize that the one that you stir, the molecules will diffuse faster than the one that is not stirred. Even you can do that with your potassium bicarbonate crystals, or it's the potassium permanganate crystals, what you call the blue that we use to wash. You realize that when you drop the crystals of the blue into the water and you don't stir, you see how long it will take to diffuse. Even if you don't take care and you don't stir, and immediately you dip your cloth into there, you see that you see some patches on even distribution of the blue in your cloth. I don't know whether any of you has experienced that before. You drop the potassium, you drop the potassium permanganate into the water, you did not stir, and you forgot and you put your clothes inside. You see that you will see patches of uneven distribution of the molecules of the blue on your shirt. And when you stir very well, you see that the molecules will be evenly distributed. And when you put your clothes inside, you see that there will be no patches of the blue that are inside. So that has to do with stirring as well. So let's look at molecules that diffuse through the cell membrane. So we have oxygen. So oxygen, carbon dioxide, and water. These are very small molecules. And so they are able to easily pass through the phospholipid bilayer in and out of the cell. Oxygen, very small. It's gas, carbon dioxide, water. Those ones are very small. And so they are able to find their way quickly through the phospholipid bilayer. Of course, oxygen is non-polar, so diffuses very quickly. Carbon dioxide, and that is as a result of the tail we spoke about. Then you have carbon dioxide, that is polar but small, and so diffuses quickly. Water is polar but also very small, so diffuses very quickly. So these, both polar and non-polar, So here we are looking at the molecular size, especially with carbon dioxide and water. They are able to diffuse quickly because of their molecular size. Oxygen is because of its non-polarity, so it's able to diffuse very quickly. And these are some practical examples of diffusion in living tissues. So for instance, when you talk of everyday activities that we can ascribe to diffusion, that one you can talk about applying your perfume. You can talk of people that release the natural gases in your classrooms and areas. Then you can talk of other real life examples of diffusion. But when we narrow it to the cell or to the tissue or to the organism, these are the real gas exchange in leaves. It's an example of diffusion where carbon dioxide enters, oxygen comes out, or in the night, oxygen goes in and carbon dioxide is released. That is example. We have water and nutrient uptake in plants. So use water moving from the soil through the rooters to the leaves for photosynthesis to take place, and the same movement of food as well. They are all then transport of sugars in plant, that is translocation of the end product of photosynthesis to the various parts. There are differences in concentration grade because the leaf that is preparing the food has higher concentration of glucose than the stem. So it is going to move along that concentration gradient to the stem as well. Respiratory gas exchange in animals is an example. Nutrient absorption in the digestive system. We realize that in your small intestine, the level of glucose is higher compared to your bloodstream. And so glucose is going to move along that concentration gradient from the small intestine through the bloodstream into the. That's why. Then you have excretion of waste products and osmoregulation in fish. So these are some examples of diffusion that takes place in plants and animals. There are a lot of examples, but these are the ones that I've outlined. You can actually read and get more. Then osmosis. Come to osmosis. So osmosis has to do with the movement of water molecules from a region of higher water potential or concentration to a region of lower water potential across a semi-permeable membrane. So with osmosis, we want to look at, so you could see that I've highlighted some points. It means that those points are very, very key. So you can define osmosis as the movement of water molecules from a dilute solution to a more concentrated solution across a semi-permeable membrane. Or you can define it as the movement of molecules from a region of lower solute concentration to a region of higher solute concentration across a semi-permeable membrane, or from a region of higher water potential to a region of lower water potential across a semi-permeable membrane. So there are three different ways by which you can be able to define osmosis, and they all meant the same thing. I believe we've taken notice of that. The pressure which develops when a solution takes up water by osmosis is called osmotic pressure. So we'll look at that in details when we move. So effect of osmosis on living tissues. So osmosis, we say, has to do with whether water moves in or water moves out. And when water moves in, there's an effect. When you lose water too, there's an effect. So we'll look at that. So the ability of a solution to cause a cell to gain or lose water is known as tonicity. So tonicity has to do with the ability of a cell to either cause a solution to gain or lose water. So tonicity, I try as much as possible to explain tonicity with comparison. So you can never, until you can arrive at a conclusion, unless you compare two things. So for instance, I cannot sit down in my house and say Michael is taller than Richmond or Abina is taller than Prince. I cannot make that conclusion. But I can only make that conclusion when I compare the two of you. So when I'm able to compare the two of you, then I can say that, okay, the comparison, Abina is taller than Prince or Evelyn is taller than what? Michael. Are we getting it? So that is tonicity. So you cannot just see one solution lying down in the lab and say, hey, this is a concentrated solution. This is a hypotonic. This is hypertonic. This is isotonic. No, you can only determine whether a solution is hypertonic, hypotonic, or isotonic when you compare with another solution. Are we getting it? So tonicity takes into account solute concentration and membrane permeability. So solute concentration, membrane permeability, but you can only ascribe when you compare. I hope you are getting it. So until you do comparison, you can never say a solution is hypertonic, hypo, or iso, unless you compare. Now, hyper means higher. So in biology, when you see hyper, it means the thing is higher. When you see hypo, it means it is lower. When you see iso, it means that it is equal. That's why we have isotopes. It means that it's equal. Hypo, it means that it is lower. So you have hypoglycemia, hypotension, low blood pressure. Then you see hyper, hypertonic, that is hypertension, high blood pressure, or hyperglycemia, high sugar level. So that is some terminology. Now, hypertonic means having a higher concentration of solid molecules than another solution. So you see that with the definition than what? Another solution. It means that you can only determine if a solution is hypertonic when you compare with another solution. I hope we are getting the explanation clear. Can I get some thumb up if you are getting the explanation clear? Okay. So that is hypertonic. Now we come to hypo, having a lower concentration of solute molecules than another solution. So hypo means it is having a lower. So it means that there are instances where one solution is hypertonic to another, but it might be hypotonic to another solution. Are we getting it? So if you take, for instance, maybe when you compare Abina and Michael, Abina is taller than Michael. But when you compare Abina to Richmond, Richmond might be taller than Abina. So in some instances, Abina is tall. But in some instances, to Abina is what? Is short or shorter. Are we getting it? So unless you compare, and when you compare, you'll be able to determine. Then we have ISO, having equal concentration of solute water. So we'll look at this in detail because they are going to help us to do some analysis. So now I believe we've gotten it in our minds now. Hypertonic, hypotonic, and isotonic, because I'll be asking, we'll look at some real life situations on this, and then we'll be able to explain. Now let's look at this. So now, based on our definition of osmosis, which has to do with the movement of molecules from a region of lower solute concentration to a region of higher solute concentration across a semi-permeable membrane. When you have a molecule, let me ask this question. When you have two different solutions, and one is hypertonic and one is hypotonic, where do you think the water will move from and why? Did you get the question well? You have two solutions. One is a hypertonic solution and the other is a hypotonic solution. Where do you think water will move from and why? I've not heard from some people. Let me see what a new. Let me see whether I'll see a new hand up. I think. Okay, let's hear from Enoch.
Hello, sir. I
Yes.
think the solution will come from the hypo solution because it has the lower concentration and the osmosis move from the lower to the higher.
Look, no, let's be clear. Lower concentration of what and higher concentration of what is very, very important in osmosis. We don't just say movement from lower to higher. We have to get the terminologies right. Lower of what and higher of what. I hope you get
lower
it.
concentration of molecules.
Of which molecules?
I think water molecules.
I want us to get the definition and the explanation right, because sometimes students, we enter into exams hall and we come out expecting that, oh, this one there I've done well, I've done this, and the result will come and you keep wondering why. But there are some basic mistakes that we make, and then at the end we tend to lose some precious marks. So I want us to get the understanding well so that you don't give away some free marks to examiners. Okay, Michael, let me hear from you.
Okay, sir. So looking about the two solution, hypertonic, hypotonic. So I mean, I'll go for hypertonic. When a hypertonic, when
So
how should I mean?
I want to say, where would the water move from? Would the water move from the hypotonic to the hyper, or hyper to hypo, and why?
Okay, so sir, I'll go for the water will move from hyper to hypo because when it is highly concentrated, it need water to neutralize it so that it won't cause it to decrease or it to smash out. So water will move from hyper to hypo.
Okay, let's hear from Abina before I clarify.
Okay. Looking at the difference between the hypertonic and hypotonic, where the hypertonic will shrink to make the hypotonic become big or swell. With a picture on the screen.
Oh, okay. Thank you. So let's understand something. Now we said that osmosis is the movement of water molecules from a region of lower concentration of solute to a region of higher concentration of solute across a semi-permeable membrane, or from a dilute solution to a more concentrated solution across a semi-permeable membrane, or from a region of lower or higher water potential to a region of what? Lower water potential. So when you pick a hypertonic solution and a hypotonic solution, the hypertonic solution has a higher concentration of solute and a lower concentration of solvent, that is water. And the hypotonic solution has a lower concentration of solute and a higher concentration of water, which is a solvent. So now, in this scenario, the movement is going to come from the hypotonic to the hypertonic, because the water in osmosis, the water will always move from where the water is higher to where the water is what? Is lower. Michael, I believe you are understanding what I mean.
Yes, sir.
Are we getting? So the water will always move from the area where the water is more to the area where the water is less, or the water will always move from the area where the concentration of the solid is lower to where the concentration of the solid or the solute is higher. I believe we get. I want us to understand this before we zoom in into the movement of the water across the various cells. So now, based on this clarification that I've given, what do you think will happen when a plant cell is placed in a hypertonic solution? What do you think? Because this is how we'll ask the question. What do you think will happen when a plant cell is placed in a hypertonic solution? Okay, Prince.
So it will shrink.
So it will shrink. Why?
Since the solution is hypertonic, it will absorb the liquid substance in the plant cell. So the plant cell will become small.
Okay, so you see that the protoplasm will shrink away from the what? From the cell wall, because it has lost a lot of water to what? To the outside. Are we getting it? Yes, Marfo. Let's hear from Marfo. Are we getting it? So then we call that condition as what? As plasmolized. Are we getting it? So we say that it is what? Plasmolized. So when you put the plant cell into a hypertonic solution, it means that the protoplasm is going to lose water to the outside. And once it loses a lot of water, the protoplasm will shrink away from the cell wall. And when that happens, we say that the plant has been what? Plasmolized. Are we getting it? And what do you think will happen when you place a plant cell into a hypotonic solution? This one I'll call Michael. Michael, what do you think will happen when you place a plant cell into a hypotonic solution?
Okay, sir. So when you place a plant cell into hypotonic solution, because the solid concentration is lower than that of the solvent, this one it won't cause any shrinkage, but rather the solution will increase.
Which solution?
So the plant is acting as a solid, and what do you call it? Let me take it again.
Okay,
When you
take it
place
again.
the plant into the hypotonic solution,
Okay.
the concentration gradient of the liquid will not cause the plant to shrink as compared to the first one, causing it to be plasmolized. By this time around, it will be more or less stable.
To be more or less stable. Okay,
Yes, sir.
let's hear from Pamela.
Okay, I think plant cell, sorry, the solute, what am I saying? It becomes turgid. The
It
plant cell
becomes
will
very
become
turgid.
turgid because, yes, because solvents from the hypotonic solution will be absorbed into the hypertonic solution. So the cell will become turgid, or the size
Okay.
will increase.
Yes. So that is C. Are we seeing C? So when you place a plant cell into a hypotonic solution, it means that the concentration of water in the plant is lower than the concentration of water in the outside solution. And so water will move into the plant. Are you getting? And when it moves into the plant, you see that it becomes, the cytoplasm will swell and swell, and you see that the cytoplasm will swell against that of the cell wall. And when that happens, we say that it is what? Turgid. But when you place it in an isotonic solution, isotonic means that the concentrations between that in the plant cell and that in the exterior are the same. And so there will be no net movement in water. So it's just going to be the same thing. Are we getting it? So look at the arrow. When it becomes plasmolized, see the arrow. You see that water is leaving the cell. Can we see? Michael, can you see the arrow? When it's plasmolized, you can see that water is leaving the cell. When it is flaccid, there's no net gain of water because the concentrations are the same. So the water enter and water leaves. Are we getting it? And when it is turgid, you can see that water is what? Entering. Are we getting it? So A is when you place the plant cell into a hypertonic solution. B is when you place it into an isotonic solution. And C is when you place it into what? A hypotonic what? Solution. So let's quickly get. Okay, Samuel. Samuel, your hand was up, or it's a legacy hand? Okay, Pamela, is your hand up for a question?
No, please.
Okay, so let's continue. Okay, so now you see that we are using turgid, flaccid, and plasmolized because it is a plant cell. This is simply because the plant cell has a cell wall, and so the cell wall offers some level of protection. But that is not the case of animal cell. When you get to animal cell, you realize that the animal cell has no cell wall. And so when you place the animal cell into a hypertonic solution, what happens is that it will lose water. And when it loses water, what will happen? It will shrink. Are we getting it? When you place the animal cell into a hypertonic solution, it will lose water. And when it loses water, it shrinks. The cell will shrink. But when you place it into a hypotonic solution, it will gain water and it will burst. Because that animal cells do not have plants cell wall against them that will protect them. But with the animal cell, the protoplasm will continue to imbibe water, and finally, when it exceeds its elastic limit, it's going to burst. So let's get the difference, especially when you put it into a hypotonic solution. So let's get that difference. Okay. Then we come to facilitated diffusion. So remember we said we have the passive transport. Remember I said we have the diffusion as well as we have facilitated diffusion. So let's see something. So larger polar molecules such as glucose and amino acid cannot diffuse across the phospholipid bilayer. Remember I said that the phospholipid bilayers, the pores are very small. And so there are some molecules that are large, they will not be able to pass through them. Also, ions such as sodium and chloride cannot pass through them. So if you have gate, assuming you have gate and your size, you are bigger than the size of the gate, there's no way you can pass through unless they break the wall or they break the door or somewhere for you to pass through. But until that is done, there's no way you are going to enter, and that is with the phospholipid bilayer. The pores are very small, and if your size is larger than the pores, you will not be able to go through. These molecules pass through protein channel instead. So you see the protein channel. Can you see where my cursor is? That is where I think it is. Is it orange or it is? Is it orange? Is it all pink? Okay, so let's accept it as. So you could see the gate. Can we see the gate? Yeah, so that is. So these molecules pass through protein channel instead. Diffusion through these channels are called facilitated diffusion. I see. So though it's the movement of molecules from a region of higher concentration to a region of lower concentration, the molecules cannot move through the phospholipid bilayer, but instead they'll pass through the bigger gate. So when it happens like that, then it is called facilitated diffusion. So assuming, let's say you have a house, you know, most houses, most gated houses, they have a smaller gate and then they have a larger gate. Are we getting the difference? So let's say human beings, definitely we human beings, we are smaller enough to pass through the smaller gate. That is diffusion. But cars cannot pass through the smaller gate. So cars will definitely use what? The bigger gate. So the cars using the bigger gate is what is termed as what? Facilitated diffusion. I believe we understand the explanation. If we get it, let me get a thumb up. Liken that to a gated house. The gated house, you have a smaller gate and then you have a bigger gate. When you are able to pass through the smaller gate, it is simple diffusion or it is diffusion. When your size is too large that you are unable to pass through the smaller gate, but you are using the bigger gate, then we are saying it is what? Facilitated what? Diffusion. So I believe that is. But all other conditions hold, whether difference in concentration gradient, whether size, molecular size, temperature, all other conditions hold. Movement of molecule is still passive, just like ordinary diffusion. The only difference is the molecules go through a protein channel instead of passing between the phospholipid. So you liken the phospholipid to the small gate, the channel to the bigger gate, and the explanation you are cool to go. So that is facilitated diffusion. So you can see the protein channels. So you could see that these molecules cannot pass through the phospholipid bilayer. It is highly impossible for them to pass through the phospholipid bilayer. So instead, they use the protein channel. And when they use the channel, that one is so simple for them. They will quickly and easily pass through, and then they are cool to go. I believe it is clear. It's clear. The image here has summarized everything for us. So let's look at this as well. So you see facilitated diffusion. You see the protein channel. So you could see that when we did the first counting, how many larger balls do you have? How many larger blue balls do we have? There are how many? Can we count? Okay, Philemon.
Please, there are 6.
There are 6. So you could see that you see the arrow pointing to the cell membrane. So you take the small pores as a phospholipid bilayer. There's no way the blue ball will be able to pass through the smaller holes. Richmond, are you getting it? There's no way the blue ball will pass through the smaller hole. Definitely, it has to use the channel gate. Are you getting it? And so let's look to the next. So can we see the movement? Are we seeing the movement? Are we seeing where the blue ball is passing? Can we see that? Yes. Then finally, you could see that there's what? Net distribution of balls. You have 3 here, then you have what? 3 here. So this is facilitated diffusion. I hope we get. And then you could see equilibrium. Equilibrium means those that are inside are equal to those that are outside. So the blue is 3 here. The red is 5 here. The red is also what? I remember the other side, the red was 11. So some will have 6 and some will have 5. But the blue, because it is even number, they are evenly distributed. 3 here, 3 here. But with the red ones, they are odd numbers. So definitely one will have 6 and one will have what one. But there will be net movement. The one will be moving intermittently. It will be moving here and then because of the difference. Because as far as it is 6, it has created a concentration gradient. So it will move here. When it moves here in 6, it will also move back here, and then it will be doing that to and fro movement. So that is. Then we look at carrier proteins. So remember we talked about carrier proteins. So transport proteins that hold onto their passengers and change their shape in the way that shuttles them across the plasma membrane. So there are some instances where shape matters. Remember, shape matters. So assuming you have a gate, and the gate, okay, Michael, let's hear from you.
Hello, sir. Sir, please, on the diffusion, talking about the facilitated diffusion, is it self-created by the tissue, or I understand that what you're talking about, that the small one will pass through the small hole, but the bigger size, unless we create a channel using the protein channel before it can.
So we don't create them. The protein channel is actually part of the cell membrane. So the cell membrane, remember we said, is made up of the phospholipid bilayer as well as protein-gated channels.
Okay, sir.
Yeah. So by convention, definitely the larger one will go and pass through the larger gate, and the smaller one will pass through the smaller gate. So it's not like that is what you want, but that is as a result of size, molecular size. I hope it is clear. So transport proteins. So there are some instances, assuming you have a round gate, will a star-shaped object be able to pass through it?
No, sir.
No. Are we getting it? So there are some instances where the shape of the molecule matters. There are some molecules, their shapes are different. And so because of the shape they have, they will not be able to pass through. And so they need to have some proteins we call carrier protein. Then the carrier protein will bind to the molecule, change the shape of the molecule so that they will have a very good shape for them to be able to pass through what? To pass through the gate in and out. So if I am star-shaped and then the gate is round, I have to be round to be able to pass through the star-shaped gate. And so I have to attach myself to a protein where that protein will change my shape to enable me to have a round shape so that I'll be able to pass through what? The round shape membrane. And that is the duty of the carrier protein. So they hold onto the passengers and change the shape in a way that shuttles them across the plasma membrane. Shuttle means moves them. Carrier proteins are specific for the substance they transport across the plasma membrane. So it means that there's a carrier protein for, let's say, glucose. There's a carrier protein for amino acid. There's a carrier protein for lactose. There's a carrier protein for this. So you don't have one carrier protein carrying glucose, amino acid, whatever. Michael, is your hand up or is a legacy hand?
So then it's legacy hand.
Okay. So you don't have one carrier protein doing all the job. Like you see one person, the person can ride a bicycle, can ride a motor, can ride a saloon car, can ride a bus, a tipper, articulator, everything. There's one person that does that. With carrier proteins, no. So there is carrier protein responsible for glucose, responsible for amino acids. So you could see that if there's an instance where carrier proteins that are responsible to carry glucose are absent, you realize that there will be protein in, there will be a deficiency in transport of glucose. And a typical example is what we'll look at that in the future. But what I want you to understand is that assuming your blood needs glucose, or your blood needs amino acid, and the amino acid is concentrated in the cells of the small intestine, and they do not get the requisite protein to be able to carry them through the walls into the plasma membrane, you realize that there will be a lot of pathology. So that is with the carrier protein. We'll look at them into details when we get to some parts of active transport. So we look at active transport. So when we talk of active transport, we are looking at transport that requires energy. So for active transport, it requires energy. Hey, our time is almost up. I hope you borrow me some 15 to 20 minutes so that I can finish up. So active transport is the movement of solute through a membrane against a concentration gradient. So you see that I mostly explain active transport with when we are moving water from the ground to the top. So assuming, let me give this real life scenario. Assuming, oh, please, can you hear me? I'm getting some poor.
Yes.
Oh, then please, can you check your network? Prince, can you check your network? Because my network is fine here. Thank you. So assuming you have water from your borehole and you have a tank on top of your story building, how do you transport the water from the borehole in the ground to the top? Anybody to help us? Okay, Philemon.
Okay. With that, you would need a water pumping machine to be able to pump the water from the borehole to the tank upstairs.
Yes. So you need a pumping machine. Without a pumping machine, there's no way the water is going to move from the ground to the tank at the top there. That is active transport. So with active transport, you are moving the substance against concentration gradient. Remember, passive transport, you are moving along concentration gradient. The thing is already moving from the high to the low. So it is natural. Once this one is higher than this, it's going to move. But with active, you are moving from the lower to the higher. And when you are moving from lower to higher, you need to apply force. And that is what? Energy in the form of what? ATP. So if I'm moving water, remember, if I'm moving the water from the ground to the pump at the top, I need to the tank at the top, I need to apply force. And that is the water pumping machine. But when the water is moving from the tank at the top to the rooms at the ground, you don't put any tank, you don't put any pump on it. It moves freely because already it has a higher concentration or higher altitude already. And so moving down is quite simple. Yes, Philemon.
Please, in this case, a typical example will be harvesting water using the rain gutters.
Okay. All right. So harvesting water using the rain gutters. Okay. So that is a very simple. So that is active transport. So you are moving the molecules against concentration gradient. When we say against concentration gradient, it means you are moving them from a higher concentration to a lower concentration. No, from a lower concentration toward a higher concentration, instead of moving from a higher concentration toward a lower concentration. And when it happens like that, you need energy, you need force, and that energy comes from ATP. So that is active transport. With passive transport, you don't need any energy, but with active transport, you need energy. So assuming two scenarios, there's one student who was forced, who doesn't know the reason why he has to go to school, or who doesn't understand why he's supposed to be in school. You see that with that particular student, you have to put a lot of pressure, put a lot of force on that student before the student will learn. And there are some students, he knows the reason why he's in school. And so he doesn't need anybody to come and tell him or she, go and learn. Immediately he knows that I'm in school for ABC really. And so he wakes up, plans his day, does this, studies and does that. But a student who doesn't understand why he's in school, he will be always sleeping unless they come and chase from the dormitory with canes before you see them moving to class. But a student who understands why he's in school, nobody has to come to the dormitory to come and chase him before he moves to class or something. So that is with active transport. They don't know why they are there. They don't know why they are supposed to come there. So you have to apply some pressure, some force, and that comes in the form of what? ATP. So active transport makes use of carrier proteins rather than channel proteins. I hope we understand. So active transport makes use of the carrier protein. It means that with active transport, they do not use the channel. They don't go through the channel. They go through the phospholipid bilayer, all right. But they have to be aided by what? By carrier proteins. And once they get the carrier proteins, they are able to enter to the various. Yeah. So let's look at the mechanism. So ATP supplies the energy for most active transport. So most of the active transport, the energy is from ATP. It does it by transferring its terminal phosphate group directly to the ATP. So you realize that ATP is what? Adenosine triphosphate. In order to release energy, it has to get rid of its terminal protein. I believe you've done this in basic genetics and cytology. You've done that. When it's released, it will release energy, the terminal protein will go. And when the terminal protein is leaving, it becomes adenosine diphosphate, which is what? ADP. So it will lose the terminal proteins, the terminal phosphate, group directly to the carrier protein. This induces a change of shape in the carrier protein, enable it to transport a solute across what? The membrane. Are you getting it? So before you release the energy, you have to release your terminal protein. Your terminal phosphate. Why do I keep saying protein? And when you release the terminal phosphate then bonds directly to the carrier protein. Once it does that, then it changes the shape. So, for instance, if you have something that is L shape, you know we've been playing bricks game. Remember bricks game? So you realize that when you add something to, when the bricks is moving and you add something to the bricks, automatically you change the whole shape of the bricks. I don't know whether you remember the guys who used to play bricks game. You see that once you add just a little molecule to the bricks, you see that you change the shape. Either you can change it to a box, you can change it to an L shape, or you can change it to even a flat shape. That is what happens there. So once the terminal protein moves and attaches to the carrier protein, definitely the shape will change. And when the shape changes, then to be able to work, to go through. So example of this is the sodium potassium pump, which exchanges sodium ions as well. So that is active. Then we have the co-transport. So with the co-transport, we have a single ATP powered pump that transports a specific solute indirectly, draw other solute into the cell by a process called so. So what you are saying is that there's co-transport. When the co-transport, when you are drawing, your intention is that I'm drawing coffee, but at the end, Amma will also follow. So when I'm intentional, so it happens in I don't know the example to use, but you see some instances where somebody will tell you that if you are able to get coffee in that particular house, it means that you will be able to get everybody to move along with you. So you see somebody, especially it's used mostly by the politicians, they will tell you that if you're able to get Michael, it means that you are most likely to get 10 other people to follow because 10 other people listens to what Michael says. Are we getting it? So that's the same thing with co-transport. When you are able to move one molecule, you end up actively transporting other molecules into what the cell as well. So for instance, a plant cell uses the gradient of hydrogen ions to drive the active transport of amino acids, sugars, and many other nutrients into the cell. So just by difference in concentration of hydrogen ions, other molecules are being what? Driven into what? Into the cell. Are we getting it? Then the diffusion of H plus back into the cell through one transport protein is combined, is coupled with the transport of sucrose into the cell against what? Concentration of gradients. Are we getting it? So that is what happens. So that is co-transport. So you move one molecule, and at the end, you are intentionally moving one molecule, but at the end, other molecules also what? Close. So that is example of co-transport. As I rightly said, the concentration gradient in H plus will either load glucose in from the photosynthesis into cells or veins of leaf. So this is mainly translocation, happens in translocation. That is when the finished product is then transported into the storage organs of the plant. So that's translocation. So this is facilitated. So example is glucose, where the protein is here, and then so this is the intestinal cell. You know, in the intestine, that's where you have the cell membrane, the small intestine, and that is where the breakdown happens. Then when the breakdown happens, so you see the transport protein. The transport protein will aid it towards move. So you see that indirectly, there's no way it is going to pass through it. Can we see the shape? There's no way this shape is going to pass through this gate. But with the aid of this transport protein, it will be able to aid it to go through. So assuming there's no transport protein for glucose, it means that the glucose molecule will not be able to enter into what? The bloodstream. I hope that is clear. And the same thing happens with lactose. So you see some people, they will tell you that they are lactose intolerant. They are lactose intolerant. It means that either they don't have the lactase enzyme to break it down, or the protein, the transport protein. Remember I told you that for transport proteins, they are specific to specific molecules. So you have transport protein for lactose, for amino acid, for glucose, and that. So it means that you lack lactose, the protein, and so they have to inject you with the lactase enzyme to be able to break the lactose down. But if you have the lactose transport protein, it means that it will be able to bind to it. When it binds to it, to be able to change the shape of it, and then to be able to, because when you look at the shape of lactose, there's no way it's going to enter. Are we getting it? But when it binds to a protein, it will change the shape, and then it will be able to enter into the intestinal cells, and then it will be broken down. So this is a table of. Okay, Richmond.
Yes, sir. I have a question.
Okay.
My question is, so does that mean that the carrier protein and transport protein, they perform the same function?
Oh yeah, it's the same. Carrier protein, transport protein, they are the same. Before you have the channel protein, that's the gated channel proteins. Are we getting it?
Yes, sir.
Okay. So just I've combined this table for you. This table will assist you to know whether this particular movement, is it passive, is it active, where does the molecules move from? Is it from a higher concentration to a lower concentration? Does it need energy? Is protein channel or protein gate needed to have? So all that we've spoken about, diffusion, osmosis, facilitated diffusion, and active transport, all of them obeys or conforms to this table. So when you get the slide, just go through this table and then you'll be fine. I hope that is clear. Our time is. Then we come to vesicle mediated transport. So there are some instances where the vesicles and vacuoles fuse with the membrane, and may utilize to release the transport of chemicals out of the cell or allow them enter. So example is what? Endocytosis and exocytosis. So as for them, they don't move alone. For the molecules to be able to move, can we see the animation? Can we see the animation? Give a thumb up if you can see the animation. Yeah, so the animation explains everything. So before the molecules will move, the molecules will first be covered in the vesicle. Then the vesicle will attach themselves to the membrane before the membrane will then finally release. And when this is happening inside, it is endocytosis. When it is moving out of the cell, it is what? Exocytosis. So exocytosis is the term that is applied when the transport is out of the cell. If it is endocytosis, it is when it's what? Inside what? The cell. So that is. So can we see endocytosis? So a typical example of endocytosis, can we see it? It's what happens with the amoeba. Please, can we see the? Can we see it? Yeah, so let me go back so that you can. Can we see? Can we see the endocytosis? I hope you can see. Yes. So example of this endocytosis is what happens to amoeba. If amoeba is taking in food, so they take the food is in vesicle and then they round their pseudopodia around it and then they are able to engulf the food. And then we have endocytosis. That's what we've and endocytosis, there are two types. We have phagocytosis and pinocytosis. Phagocytosis is when the food is in a form of solid, and pino is when it is in a form of what? Liquid. So are we getting the difference? If it is phagocytosis, it is solid. If it's pinocytosis, it means that it is what? Liquid. So all these are. Then we come to exocytosis. That is when, can we see the exocytosis? When it is released outside. So when it's released outside, it is exocytosis. I hope we can see. Yeah, so that brings us to the end of the lecture. So as usual, I'll quickly share the slides with your classmates immediately so that I want to do it at a very quick pace so that now that once we've gone through the lecture, I believe the information is fresh in your mind. So if you go through the slides, revising is quite easier and simple. So I'll quickly share the slides with your rep so that you can send it onto your page. So thank you very much for your attention. In case there's any question, I've used about 8 minutes of your time. In case there's any further question, please kindly speak to your yes.
Please, you called me, but I was having a problem with my network. That's why I couldn't speak. I'm really sorry.
You said I called you.
You asked a question that you called my name that I'm supposed to answer, but I was having a problem with my network. So when I was speaking, it was not coming.
Oh, okay. Okay, that's fine. So Richmond, right? So that's somewhere, right?
Yes, sir.
Okay. So I'll send the slides to Richmond. Is Richmond here? Yeah, I didn't
yes,
hear.
sir.
Hey, it's like today you are silent. So why?
I'm Kujo Richmond.
Kujo Richmond. Where is
Yeah.
Jomo? Where is Richmond Jomo?
Jomo is absent.
Because I didn't. Okay, so tell him I've sent the slides to his email so that he can share with you guys. Well, I think I only have his email.
Okay, sir.
So okay, so just tell him I've sent the slides to him. Okay. So have a nice evening. We'll meet tomorrow at 7 a.m. Bye.
Bye, sir.